All right, this video is going to focus on antibodies, which are made as part of the humoral arm or humoral branch of our specific immune response. Okay, so cute little joke here to kick us off. Why don't ants get sick? Because they have little antibodies. Oh, it's so cute. Okay, so we are focusing on our humoral response, okay, that first arm or branch of our specific immune response. And so before we really get into like B cell activation and B cell differentiation, let's focus on the outcome, the antibodies, because it's really important that you understand how antibodies work. And so the big goal of antibodies is that they are going to be what binds to the antigen. Okay, so the antigen is that the molecule that, is recognized as foreign by the body. Okay, so that's what the antibodies are going to bind to, and in doing so, it targets them for destruction. So, kind of going bigger picture, where do we get antibodies? B cells. Okay, so B cells are going to mature in the bone marrow, and that's when they're going to migrate over to our lymphoid tissues where they complete their development. And so you can see in this picture here, let's see, am I on the right screen so that you can see it? There we go. Sometimes antibodies are membrane bound, and they are displayed on the surface of that B cell. Okay, and you'll learn later that not all antibodies are confined to a B cell, but think of these antibodies as like receptors on the B cell. They are going to be looking for their specific pathogen. Okay, so that way we have B cells looking specifically for E. coli, B cells looking specifically for Salmonella, B cells looking specifically for COVID, or whatever it is that you've been exposed to in your lifetime. We've got B cells on the lookout for each of those pathogens. So let's talk a little bit about antibody structure. So antibodies have this Y shape, and I've put a different picture here than what's in your PowerPoint that I posted for the class, just to kind of give you a different visual. And I like the contrast on this picture here. But notice it's a Y shape made up of 4 chains, polypeptide chains. These are proteins, so again, they're made up of amino acids. And so you'll notice that in the middle, right, of this Y are these 2 longer chains. So we call those 2 inner chains, the long ones, we call them the heavy chains, right? They're bigger, they would be heavier. And the 2 chains on the outside, we call those the light chains, okay? So 4 polypeptide chains, 2 light chains, and 2 heavy chains. But you'll notice that part of the heavy chains are a dark maroon, and part of the light chains are also a shade of maroon. Okay, these maroon regions are called the constant regions. Okay, and they are going to have the same shape, made up of the same amino acids, which is in contrast to these yellow goldish regions, which are the variable regions. Because remember, our specific immune response is specific to different pathogens. So we need antibodies for E. coli, but we need different ones for Salmonella, right? E. coli and Salmonella are 2 different pathogens. They have different looks, they have different antigens. And so we need the antibodies to be specific for each pathogen. And so it's this variable region that is going to vary in shape based on the pathogen that it is specific for. So in this one, you see the little oval looking shape of the variable region. And so this antibody is going to recognize the specific pathogen that has this specific shaped antigen. Okay, so different antibodies will have different shapes here in this variable region. Again, that is specific for its pathogen that it's looking for. And then, as I just mentioned, some antibodies are bound to the surface of B cells. And so you can see here at the bottom, that's where the antibody would bind to the B cell. Okay. So in this variable region, okay, that's going to be what binds to the antigen. So again, just kind of really reinforcing the concept here of complementary shapes. The shape of the antigen is going to match the shape of this variable region of the antibody. Now, oftentimes, the antigen, what the body recognizes as foreign, is a pretty large structure compared to itty-bitty, teeny tiny antibodies. And so an antibody can only bind to certain parts of the large antigen, okay? So the specific part of the antigen that binds to the antibody is called an epitope. Okay, now this is a lot of terminology, so I created this picture down here on the bottom. So this is going to be a bacterial pathogen, right, with our monotrichous flagella. You'll know I love flagella. So the bacteria is the pathogen. Now maybe with this bacterial pathogen, our immune system recognizes the big old flagella as the foreign molecule. Okay, that's how it knows that we're infected with this bacteria, because it detects the presence of its flagella, which is different than eukaryotic flagella. But look how big that flagella is compared to one antibody. And this is definitely not to scale y'all. I just tried to create this using PowerPoint, so that was as small of an antibody as I could make. But you get the point that the antibody is much smaller than this big old flagella, which is the antigen. And so what the antibodies will actually do is bind to specific molecules that make up the flagella, okay? And so if the flagella has these little circles on it, okay, then that might be specifically what the antibodies can bind to. So the circles would be the epitope, the flagella is the antigen, the bacteria is the pathogen, okay? So another way of saying this is that epitopes are part of the antigen, okay? Epitopes are part of the larger antigen, and that's what our antibodies are going to bind to. Now, because you definitely need more vocabulary in your life, right? There's no such thing as too much vocabulary. Let's introduce another detail, okay? So we just said that the epitope is part of the antigen, which means it's on the pathogen. Well, we said that the antibody has this variable region that is going to vary in shape based on its specific epitope. And so we call that specific spot where the epitope is going to bind, we call that the paratope. So the paratope is on the antibody, which is part of our human immune system. Okay, so the paratope of the antibody binds to the epitope, which is part of the antigen, which is on the pathogen. Let that sink in for just a second. Okay, so as I said, each antibody is going to be specific for its unique target, its pathogen. And so as you can see in this picture, it's just a visual of what I've been saying. We have two different antibodies here. Notice the specific shape of their paratope is different, right? This green one has more of a triangular shape, whereas this second antibody on the right, I don't know, it's got like more squarishy shapes here in the paratope. So you'll notice if these are different antigens that our body or our immune system has been exposed to in the past, notice only one of those antigens is going to be able to bind to the left antibody, okay? On the right, that antibody is going to bind to its own specific antigen, okay? And so notice, again, let me get my pen to just mark this real quick. So you'll notice that, oops, I clicked on highlighter. It's not going to do me very well. Only this part is actually going to fit into the paratope, right? Right here. Okay, not the entire thing. So that's going to be our little epitope. Okay, that's going to bind to the paratope on our antibody. Same thing. This is going to be the part that fits into the paratope. Okay. But again, each type of antibody is unique to its own pathogen. Now, some pathogens are going to have, because maybe they're larger, like a virus isn't terribly large, but maybe a virus here has multiple spike proteins on its surface. That's very plausible. And so maybe our immune system wants to recognize as many of those spikes as possible, right? We do not want this virus to evade detection. So we can create antibodies against multiple antigens, okay? Which means there's multiple epitopes. So on this example, okay, we have a virus with an icosahedral capsid, and notice it's got different spike proteins coming off of its surface, okay? And so this capsid might be the big antigen, which is what our immune system is detecting. But notice on the capsid, again, multiple spike proteins. So the purple one serves as one epitope, the orange one serves as a second epitope, and then this green one over here serves as a third epitope. Okay, so we have 3 different epitopes on this one big antigen. Okay, so let me kind of clarify that, because I think I misspoke a few minutes ago. In this case, we're considering the capsid as one antigen. Okay, and so the capsid antigen has multiple epitopes. All right, so I'm going to kind of wrap up this video with kind of how do we get such, diverse antibodies. So it's kind of a complicated process, and I only put one slide in here. But what is going to be important to understand is that it's this variable region, right, that's really varying. And then we're going to have these constant regions, okay? So the 2 paratopes on a single antibody must be identical, right? And so, hold on, y'all. So if this purple antibody here is made by a specific B cell, it can mass produce lots of purple antibodies and only purple antibodies, okay? A B cell that has antibodies on its surface can only make that type of antibody. A different B cell could make this antibody on the right, because its paratope is a different shape, right? So antibodies are proteins, and what determines the shape or structure of a protein, right? The DNA, right? Because DNA makes RNA, and RNA is used to make protein. So at the DNA level is going to be where this diverse array of antibodies comes from, okay? Now, I'll be honest, it's a pretty complex process, and so just wanting to give you a very brief dabble in this. I'm not going to test you on DNA rearrangement, because that's essentially what's happening. As you can read on your slide here, the antibody genes in an immature B cell, which means it hasn't been activated yet, there are 40 variable segments, 5 joining segments, right? And a constant segment that is going to make up the variable region of a light chain. Okay, the heavy chain is a little bit different, but that's a lot of segments, right? There's a lot of DNA and nucleotides involved in this. And so essentially, what's going to happen is we have enzymes. And again, I'm not testing you on these details. I just kind of want to give you a taste of why and how we have so many different types of antibodies. We have an enzyme called DNA recombinase that is randomly going to excise or remove a lot of those segments out of the gene. And in doing so, they're going to splice. Okay, remember that's where we talked about introns and exons. They're going to splice one variable segment and one joining segment. All but one variable segment and all but one joining segment will be spliced out. And so with recombination and splicing, there's over 1 million possible variable and joining combinations. And so each differentiated B cell in the human body is going to have a very unique variable chain. Whereas that constant region is pretty constant, is going to be the same for all classes of antibodies. So once all this, rearranging has occurred, the antibody's genetic code is going to be permanent for that particular B cell. And so all antibodies produced by that specific B cell will now have that specific variable region and paratope, which will be unique to one particular pathogen. Okay, I know it's complex. Please don't spend a bunch of time re-listening to that section and memorizing it. It's a lot of detailed genetics, really. But people tend to ask, well, how do we get such diversity in antibodies? And so I just kind of wanted to let you know it's because of what happens after we do transcription and RNA processing with splicing, trying to kind of connect the dots back to some Unit 1 material, that splicing process is going to help create the genetic diversity of our antibodies. All right, moving right along now. And it's just a different picture kind of showing you these things, okay? Genes encoding the variable regions, different RNA transcripts are going to be made from the same gene because of that RNA processing. And based on the transcripts that are made, right, each piece of RNA is going to code for different amino acids. And so that's why it's going to vary. Okay. Why is it not changing slides? There we go. All right, so as we said, once that B cell has its specific DNA, it is going to mass produce the same kind of antibody over and over again. And as we said, some of those antibodies stay on the surface of B cells, whereas others are going to be released once the B cell is activated and differentiates into a plasma cell, because those plasma cells are going to mass produce antibodies that get released into the body to go circulate and try and round up all of those pathogens and target them for destruction. Okay, so some cool facts for you down here. I'm not going to test you on these numbers. But it's really cool how quickly we can mass produce antibodies. So because DNA rearrangement is a random process, right? We need to make sure that our immune system is only producing, that our bodies are only producing B cells that are going to make antibodies against pathogens, not against ourselves. So we essentially have this vetting process where in the bone marrow, where the B cells are maturing, there's a lot of self-proteins present in that bone marrow. So as new B cells are made, they're essentially tested. They are exposed to different self-proteins, which they should essentially ignore. And if they pass the test and they ignore all the correct proteins, they are released out into the body to go do their job. But if a new B cell attacks a self cell, that B cell is targeted for destruction because that would be targeting our own body, and that would be bad. So there is like a vetting process to make sure that our immune cells only recognize non-self pathogens. All right, we're going to go ahead and end this video here. We're about to get into the different classes of antibodies, and I'd like to make a video just for that. So go ahead and let me know if you have any questions about this content.